Good evening. Thank you for coming to our Illinois RiverWatch Winter Speaker Series. This is our first event of the year. And this is also a joint event. So for those of you who are on Zoom, we also have a live audience here tonight who are here for the National Great Rivers Research and Education Center's Neighbor Night event. So that's an event we have on site here generally once a month, skip a few here and there. My name is Danielle Hake. I am the director of Illinois RiverWatch and a stream ecologist here at NGREC. And so I'm here to introduce our speaker for the evening. But before I do, for those who are online, tonight we're going to ask that you stay muted the entire presentation. If you have a question, please ask it in the chat, and then it will be asked here live. If there's not really a good system set up for you to ask questions live and everybody to be able to hear that. So just ask in the chat and we'll make sure that the questions get answered. And with that, I would like to introduce our speaker for this evening. Hannah Griffis is the RiverWatch technician and volunteer coordinator. She's been working with me here for a couple of years now and don't know what I'd do without her. We started this process of working with freshwater mussels a couple of years ago, and she's been helping keep that process flowing just like everything else RiverWatch. So for that, then I'll turn it over to Hannah so that she can talk about what we've been doing to look at the biodiversity of freshwater mussels in the Upper Sangamon River.
All right, thank you, Danielle. Now let me see how to get the presentation to work. Okay, so to start out, just an outline of what we are going to be doing tonight. I'm going to start out by giving you guys some information on freshwater mussels in case you aren't very familiar with them. And then I'll talk about how our project got started. And then I'll run through the methods that we use for this study, followed by our very, very beginning of results. And then I'll talk about the future of the project, and then we will have our short film premiere after that, and then we'll have questions after the film. So to start out, what is a freshwater mussel? So a freshwater mussel is a bivalve mollusk. So that means that it has two shells. That's what the bivalve stands for. And then mollusks all have a hard shell with a soft body inside. And these guys are very similar to their marine counterparts, but they're located in our freshwater rivers and streams. And most of the mussels that we have in Illinois are from the family Unionidae. And mussels are really important for our rivers and streams. They're filter feeders, and they filter gallons of water every single day, which removes impurities and helps keep the water cleaner. They also have a really interesting reproduction cycle. They have a parasitic larval stage. So you can kind of see in the bottom picture how the cycle works. So after the female's eggs are fertilized, the young will stay inside the female until they are ready to be released. And the young are also called glaucidia. That is what the larval form of a mussel is called. And the female will have different sorts of methods that it uses to attract its host. And the host is usually a fish. And once it attracts the fish, it will basically, most of the time, just spray out the larva onto the fish and then hope that the larva land on or near the fish gills. And then the larva will hang out on the fish gills for a while until they reach the juvenile stage. And then once they reach the juvenile stage, they just kind of drop off and start their new lives, ready to become adults. And the United States is a hotspot for mussels. We have a lot of diversity of freshwater mussels. And in the Sangamon River, which is what we are going to be focusing on today, there were historically 48 species of freshwater mussels. But in surveys conducted by the INHS, the Illinois Natural History Survey, in 2009 through 2011, they only found 28 species. And that's kind of what we're seeing everywhere in the US is mussel species, both their numbers and their diversity is declining. And so now that you know a little bit about freshwater mussels and why they're important, I'm going to talk about how our project got started. And to start that off, in case you guys are unfamiliar with our program, I'm going to give you a little bit of background on that. So Illinois RiverWatch Network is a statewide citizen science or community science water monitoring program. And if you haven't heard the term citizen science or community science, it's basically where everyday people can go out and help contribute data to scientific projects. So you don't have to have any sort of scientific background, and we get all these volunteers to help contribute scientific data. And so our mission for Illinois RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. And just a little bit of background about RiverWatch. The program started in 1995 by the Illinois Department of Natural Resources, and it was started as part of the EcoWatch network. And the EcoWatch network had a bunch of different programs under it. So it included things like Forest Watch and Prairie Watch and Urban Watch and RiverWatch. And then in 2004, IDNR lost funding for these programs. And to this day, RiverWatch is the only one that's still going on. And in 2006, the National Great Rivers Research and Education Center, NGREC, adopted RiverWatch and still runs it today. So for those of you who are virtual, this is a picture of NGREC, and we are located right outside of St. Louis, but on the Illinois side. And you can see we have this really nice green roof, and we're right along the river. So if you guys are ever in the area, you should come visit. And for those of you who are here, you didn't get to go up on the roof tonight because it's dark. So if you haven't been up there, I really suggest that you go up and see it. And it does look different than this picture because all of our prairie plants are all there now instead of just the short green grass that's going on in the picture. So RiverWatch has a lot of different projects that we have going on, and this is just a few of them. So our main thing that we've been doing for over 25 years now is our macroinvertebrate monitoring. So this involves collecting water bugs to determine water quality. We also have a temperature monitoring program, which involves putting like a data logger out in the water, and it just stays out there and collects continuous data. We also introduced a winter chloride monitoring program in 2021, and that has been gaining traction since we've introduced it. And then finally, what brings us here tonight is we also have a mussel monitoring program. And from this program, the Upper Sangamon River Conservancy had been monitoring mussels in the Sangamon for over 10 years at the time, at the beginning of our project, so 12 years now. And they reached out to us and said, hey, we've been doing this monitoring and we'd like to get more information, better information. We'd like to start tagging the mussels. So once they reached out to us, really Danielle took the lead on this, but there are 5 partners altogether for this project. So that includes Illinois RiverWatch, the Upper Sangamon River Conservancy, Illinois Natural History Survey, Missouri Botanical Garden, and the St. Louis Zoo. And this project was funded by a grant from the Living Earth Collaborative. And for this project, there are 5 main questions that we're kind of looking at answering. We want to know how many mussels are in the Sangamon and what species are there. We want to know how do mussels move in the river, and this isn't to say how do they physically use their foot to move. This is to say, do they go upstream, downstream, that kind of thing. We want to know what kind of stressors are affecting the mussels. We want to know if the genetic makeup of mussels changes along the river. And we also want to know how their size, shape, and growth rate vary among the mussels. And so to do this, we had 8 study sites that you can see here, and they're all along the Sangamon River. And this was done in central Illinois near Champaign. And so from the most upstream site to the downstream site, that is 42 miles of river between them. And the project is set to go for 10 years. And so far we are 2 years into the project. And we only monitor 4 sites every year. So all of these sites have only been monitored once so far. And so when we do the monitoring, this, as I said before, is a citizen science project. So we have lots of volunteers that help out. And our first step is to do the setup. So we have 10 transects set up, and you can see that's what they're setting up in this photo. So those orange poles are the transects being set up. And then we also have an upstream and a downstream area. So only the mussels that we find within those transects are the ones that we tag, because we have a specific amount of space, time, and effort put into there so it can be comparable. And the upstream and downstream sections will also give us data, but we are not tagging those mussels. And these transects will also help us learn about how the mussels are moving up and down river. So when we go back and find them, we try to set up the transects in the same spot so we can see how they're moving. And then we give a mini training to the volunteers who are there. Many of them have never done anything like this. So we don't expect them to know anything before they come in. So we teach them everything they need to know during the events. So in this picture here, you can see Sarah Douglas is a malacologist from the INHS, which is just a fancy way to say she knows a lot about mussels. And she's giving, I think in this video or in this picture, they've already collected the mussels. So she's probably giving information on how to identify them. But a lot of the events, she kind of leads the training. But then the other partners also help out. And then once people are trained, we do the collecting. So this is how you collect mussels. You literally just sit on your hands and knees and dig around in the sediment until you find one. And you can see in the picture, these people have these yellow bags, and those are just like mesh laundry bags. And that's what we keep the mussels in because it allows water to flow in. And all the bags and the tubs that you see are labeled for the transect, so we can keep track of what came from where, and then things can go back in the same place as well. Next, the mussels are tagged. So I believe the tags are vinyl tags, and we just put them on with super glue. Since it's just going on their shells, it doesn't harm them in any way. And we tag both sides of the mussel so that one, if a tag falls off, we have a backup. And two, if we find dead animals and it's just like half the shell, we still have a tag. And then we move on to identification and measurements. And so at this stage, we are taking measurements on the mass, so how much does the mussel weigh, the depth, so that's like how thick the mussel is, and then the length of the mussel. And then for part of the study, as I mentioned before, one of the study questions is looking at size and shape and growth rate. So the way we're doing that is through a study of shape, which is called morphometrics. So we're taking pictures of all of the tagged mussels, and this is the setup we have for that. So we have two GoPro cameras, one from the top and one from the side. And then you'll see some pictures of this later, how they turn out. The cameras are set off to take pictures at the same time. And then all the mussels are positioned in the same position, the same height from the camera, so that when we have all these pictures, we can go back and see how the shapes change between individuals. And then there's also a genetics component to the study. So again, Sarah Douglas is the one who leads the genetics portion, at least taking the samples. So you can see that's what she's doing here, is just a small tissue sample. And there's two mussels that we focus on for the genetics, and that's going to be the round pig toe and the pistol grip. So we don't do this for all of them, it's just two species. And then finally, once all that data is taken, the mussels finally get to go home. So we try to put them back in the same place that we found them, so they all go back to the transects that they were picked up. And then we also take some habitat data. So this includes water chemistry, so things like dissolved oxygen or pH. And then we also, as I mentioned earlier with the temperature monitoring program, we also put one of those data loggers in, and it stays throughout a full year before we pick it up. And then we also grab, I think, 4 sediment samples from the transects, a water sample, and then we do some light measurements to see how much shade and how much light is coming through in the site. So some of the common mussels that we find, some of the common mussels that we find in the Sangamon are listed here with the numbers of how many we found of each one. And then you can also see how the morphometric photos turned out. I did crop them. So if you notice that some of them aren't positioned exactly the same, it's because I cropped them so you could see the smaller ones better. And you can see that mussels have a lot of diversity. They have a lot of different shapes and sizes going on. So throughout the 2 years that we've done this, we have collected 2,796 mussels. And of those mussels, 1,411 of them were tagged, and they included 19 species. So in these graphs, the sites are listed from upstream to downstream. And on the top one, species richness is just talking about how many species there are. So that's a measure of diversity for us. And the abundance is the total count of mussels we got at each site. And both of these graphs are just looking at mussels that we tagged. So it's not including the ones that were found in the upstream, downstream sections. And if you look at the graphs, you can notice that there's a really high diversity and a high abundance at our most upstream site. And then not as dramatic, but there is a higher amount of diversity located in our downstream sites as well. And for those sites, in the upstream site, the Sangamon River Forest Preserve, there were 3 species that we found that were not found at any of the other sites. So that includes the spike, the fat mucket, and the monkey face. And then for the bottom 3 most downstream sites, there was 2 species that were not found in any of the other sites that were upstream. And that is the pink paper shell and the maple leaf. And then for our sediment samples that we took, we basically shook them through a bunch of different sieves to sort out the different sizes of sediment that were at each site. And so some of the sites were dominated by gravel, which is the blue colored bar. And some of the sites were dominated by coarse and medium sand, which is the gray and yellow. And then some of the sites like Shady Rest in the Sangamon River Forest Preserve are a mix of sediments and don't have a dominant type. So that's really all to say that we didn't really find a pattern in upstream versus downstream sediment makeup. And then for the future of this study, like I said, we're just starting. So this is just the very beginning of the data. Currently, our genetic and our morphometrics analysis are in progress. So our genetics are being done at Missouri Botanical Garden, and we do have an intern who's working on that. So we're hoping that that is all processed by the summer this year. And then our morphometrics analysis is also in the works, but it is very time consuming. And we still have 8 more years of this study. And so a lot of our questions can't be answered yet because you can't really tell population levels or movement or growth rates until you go back for another year and get more data from the same sites. And so we have a lot more data coming, and we're looking forward to the next 8 years of working on this project. I also went through this very fast, and we are now at the video component. I don't know, Danielle, do you want to take questions now and wait on the video?
Take questions.
Okay, so we'll do some questions before the video and plan on doing the video at 7:30, and then we'll take more questions after the video. So if you're online, you can answer or put your questions in the chat and we will have them read out loud. And if you are in person, you can raise your hand.
I'll go ahead. There's already one in the chat.
Okay.
Was the amount of volunteers and the time spent sampling the same for each site?
Okay, so the question was, did we have the same number of volunteers and the same time spent sampling at each site? So did we have the same number of volunteers? The answer is no. Some sites were more receptive to having a large number of volunteers show up, whereas other sites, it was better to have a smaller amount because there was less parking or less access or things like that. But every transect did have the same amount of time spent searching for mussels. So we normally assign 3 people to search each transect for 30 minutes. Occasionally it would be 2 people for 45 minutes, but all of the transects had the same amount of effort put into searching.
Okay, a question, where did the mussels get their names?
I don't know. Scientists do a lot of weird naming stuff. I think a lot of them are just based on what they look like. For example, the pimpleback has pimples on it, and the 3-horned wartyback has those 3 bumps along its back. So I think it's just a lot of, it kind of looks like this thing, so I'm going to call it that. And then a lot of, I found it first, I get to make the name.
Okay.
There's a question in the back. I'll take your question next.
Are you limiting the genetic testing just so it's two varieties, not data?
I think it is a data thing because I think genetics, Sarah could answer this better. I think she's going to be on later. But I do think genetics is an expensive test. It's also, yeah, we have to have enough mussels that we're collecting. So like some of the more rare mussels wouldn't provide enough data for genetic testing, because I think we did have a different species set out at the beginning and we had to switch it because we weren't finding enough of them.
Yeah, we wanted things that we could find at multiple locations. So, you know, we didn't want something that was like, we had, like, there's those species that are only at the upstream site or only at the downstream 3. We wanted things that would be likely to be found, and we were hoping for at least 10 of each at a location, or at all 8 locations. And so that's kind of, but we also wanted to focus on things that are maybe a little bit, you know, that aren't the most common species because they are less likely to need help, so to speak. All right, so we've got a question. What are the squared measurements? I'm not sure what that was, if that's
the transects?
I possibly, yeah, and metric or English. So yeah, I don't know if there was anything else that was numeric.
If you could clarify that question in
Yeah.
the chat. There's a question in the audience.
Yeah, please repeat the questions from the public.
Yeah, yes, I realize I
Okay.
messed it up. Yeah.
Okay, my question was, and she alluded to it, was, you know, you said that you found the maple leaf and the other one on the downstream side, and there was a couple that you found on the upstream side that were they normally throughout, or do you know that? I mean, or is there something special about the upstream for this species and the downstream for this species?
So the question was, why were some of the species found in the upstream versus downstream, and vice versa? So I think we're not quite at that answer yet. It could be, yeah, we know it's not sediment. That's as far as we've gotten. We haven't really looked at the chemistry data yet. There could be a difference in the chemistry makeup. It could be based on what is happening around the sites. So there's not too many cities around the sites, but right in the middle of, like, we have 4 sites above and 4 sites below is a medium-sized town. So that could have something to do with it. But at this point, we're not at the point that we can actually answer that question.
I was wondering, because, you know, you said at the beginning there were like 49 species that they had found before, and when you're checking, and there's 19.
Right.
You know, something like that, 19. And so I didn't know whether something there, you know,
Yeah, so he was just mentioning about how historically there's only been 48 species, but we really only found 19. I don't know what INHS's methods were when they went out and did their surveys. So I don't know if ours used similar, I'm sure they were similar methods, but I don't know what the differences is. So that could be part of it. Theirs may have been more intensive. But we have just seen a lot of decrease in population size and diversity across the whole country. So it wouldn't surprise me if we only have 20 species now. I'll take a online question and then I'll go back to.
So I've got a clarification on the other question. When the mussels are photographed, the white background has the black line squares.
Oh, okay.
Which size of those? So those are
Yeah, so the squares for the morphometric pictures are 1 inch, and we use that as a grid for the mussels, but actually we ended up using the ruler more for the pictures than the grid. Yeah.
How long ago did they determine there were 48 species here?
Oh, so the question was how long ago was it that they determined that there was 48 species?
There were 48 species in the Sangamon River in this area.
Yeah, so I was gonna say like 60s, 50s is probably when that was done.
And how do they move? Physically move?
Well, they have, so the question was, how do they move? So they have their foot that they can move across sediment with. So they're pretty slow movers, but I also would assume that they can get dislodged like during bad weather and kind of like float downstream and stuff like that. Yeah.
How much water can they filter in a day or an hour? How do you measure that?
So the question was, how much can they filter in a day? I don't know how that's measured. From the website that I was looking at earlier when I got that, when I was getting that information, they did say that they filter multiple gallons a day, but that's the extent to my knowledge.
So in theory, more mussels would mean cleaner water.
Yes, more mussels would mean cleaner water.
Okay, so I got some more questions
Yes.
here. Let's see. How do the mussel numbers and diversity in the Upper Sangamon compare to other rivers you have surveyed?
So the question was, how do the numbers that we found in the Sangamon compare to the numbers that we have also surveyed? From Illinois RiverWatch, we have not done too many other surveys. We have had some other citizen scientists who have done the mussel monitoring and collected data. I think that would be another good question for Sarah if she logs on later.
Were any invasive species found in the river?
Were any invasive species found? I know for sure that we saw some Asian clam. I don't think we found any zebra mussels, and outside of that, we weren't really looking for invasive species, so that's all I can speak on.
Are your species counts live mussels only or all shells found?
We are looking at live mussels only. Yep.
Right. Any tips for volunteer recruitment and retention?
Well, so tips for volunteer recruitment and retention. That's kind of what we're doing now, is giving a presentation, and you guys are also going to see a video featuring the citizen science perspective. Yeah, provide food. I feel like it's really good. I feel like a lot of people who show up to these kind of events don't know what they're getting involved with. So the more information you can provide and like first on look of what is happening, the better.
Do you need a DNR permit to collect freshwater mussels?
Yes, you do need a permit from the IDNR. You need, I think, a general collecting permit for sure. And then there is also a threatened and endangered species permit, because a lot of the mussels are threatened or endangered, especially if you don't know what you're looking at beforehand. If you're able to tell which ones are endangered and put them back, you might not need that permit, but for the most part, you do. Let's go ahead.
It's only for the, like, the leaders need to get permits, not at each volunteer.
Yeah. So most of the time for projects, just the leader has to have those permits. Let's go ahead and do the video now. So the video is called Mussel Grubbing, a Citizen Science Treasure Hunt, and it is funded by the Consortium of Universities for the Advancement of Hydrologic Science from their Let's Talk About Water grant. And this grant is aimed at bringing information about the importance of water out to the public. And so I just gave you a whole presentation about the science side of things. So in this video, you're going to get the view more from the citizen science perspective. And this video was all filmed in fall of 2022. And Danielle will be sharing the video, so we just need a second for setup.
I have a special knack for finding things. When I did a whole 12 weeks of discovering what I wanted to do, the thing at the top of my list was treasure hunting and finding things.
Some of the nicknames that come about is mussler and mussel grubber. I think more technically it could be called amateur malacologist.
Sometimes we call it pollywogging, sometimes we call it grubbing, and you're just trying to go on this treasure hunt for finding freshwater mussels. So today we're out here to do some freshwater mussel surveys. We're trying to find out how many mussels are living in the Upper Sangamon River and working to better understand how the mussels are growing and how their populations are doing here.
My experience doing the mussel surveys is new. Started this year and went to all 4 events. It was a blast. Coming out here on my first day, I was not sure what to expect. I was laughing almost the whole time and deep in the silt and the rocks trying to get mussels, and initially I just thought I'm gonna find some mussels in the water and bring them back so that other people can do all of the processing and categorization of them, but I was able to be involved in that process, which was really exciting for me. I also learned about different mussels. Guess I didn't really come with any kind of expectation that there would be a specific kind, but there were so many more than I could have anticipated.
It's important to study freshwater mussels because they can help us determine if our water is healthy, if our waterways are healthy, or rivers, then we're going to have healthy people. When we kind of connect those dots of healthy animals in the river, a healthy river, then we can have healthy people. At this site in particular, we have a lot of really cool mussels. One of the things that we've also found here is some of the more rare mussels, one of those being the monkey face. Currently it's a state threatened species, and we had a couple other rare species like the fluted shell that was found. We don't find too many of those. The other neat thing too about this site is there are a lot of mussels, and so we've got a nice bed essentially. A lot of times mussels are really patchy, but they're just found throughout this section of the river here. That tells us that these animals are happy, that they're able to thrive here.
The first mussel that I found was a pimpleback. I was very excited because it took a while to find, and I was so focused on finding one, and I did. Yeah.
One of the goals of citizen science with the Upper Sangamon River Conservancy is to offer opportunities to young people who might then take that enthusiasm that they have out here for the surveys into a more long-term goal of theirs. And the citizen science works very well for both getting precise information that we needed about the mussels and allowing great opportunities for citizen scientists of all ages and experience to be able to make meaningful contribution to science.
In this particular survey, we had scouts out and they pulled up a super rare mussel we have now in Illinois called spike. It's state endangered. The last record that we had live for the Sangamon River Basin was in 1991. And then in 2022, a live individual that was fairly young, looked healthy, and that was just a fantastic record. Being able to see that helps me think of, future ideas. One of the great things about citizen science is that anyone can participate.
Just in this month so far, we've had volunteers who are 5 years old. We've had volunteers in their 70s. We've had volunteers who are legally blind. You can't usually see the mussels anyway. You're using your hands to find them. So being blind wasn't necessarily a hindrance to participating in this citizen science project. And we also have a lot of diversity of backgrounds. You know, we have people with different educational backgrounds, different racial backgrounds, different aspects of human diversity are being well represented in our quest to understand the mussel diversity.
One of my biggest passions is to bring people outside, and in particular, Black and brown people outside, and make it less scary of a experience and a really exciting opportunity. So I think that citizen science does that inherently, where you're able to come out, things are structured, you learn about what things are, what things to look out for, and then you are in it. I think if someone is feeling nervous to come out and participate, it's totally understandable and no need. You can always leave if it's not for you. For me, I would just say, do it. Try it. Try something new. Go for it.
Bruce earlier as well.
Okay, so Sarah Douglas and Bruce Colravy are in the chat now. And they are two of the major partners that we were working with on the project. Will you check and see if there's any other people? Both of them were featured in the video. So Sarah Douglas is the malacologist with INHS that we worked with. So she can definitely answer any more in-depth mussel questions. And Bruce Colravy is, I believe he's the volunteer coordinator for Upper Sangamon River Conservancy. And he is also the one who came to us to begin this project.
Yeah, and they're both on screen now, and they were both featured in the video as well. Yeah.
Go ahead.
Okay, yeah, I can do it.
Okay.
So we have a couple of questions from before. I see great decline in mussels in many rivers and streams. How serious is this? What's the impact for the future?
Okay, so the question was, with all the decline in mussels that are happening, how serious is it, and what is the impact on the future? So I don't have a lot of background on this, but from what I have learned, it seems pretty serious, because it seems like a lot of the reports are saying that there's less than 50% of the species in specific watersheds that are located there. And while losing biodiversity in any sort of habitat is bad, so losing these animals is decreasing the biodiversity, we're also losing them as filter feeders. So we're losing that filter system that is happening naturally in the river, which causes more pollutants to just sit in the river. Wait, I'm going to take an in-person question.
What pollutants are the biggest killers?
So the question is, what pollutants are the biggest killers of the mussels? So sure, Sarah, do you want to answer the question?
Sure. Well, so you can kind of
hold on, we have to figure out our in-person.
Oh.
I think we're good now.
Okay. There's two things you can consider in terms of pollutants, where we have some that are acute, where something will kill them, pretty immediately if there's a big spill, for example. We've seen that with cattle or hog spills, usually some sort of manure, so you get a lot of nutrient input, like really quickly. And it just essentially drops the DO in the water, and then mussels will die from that, or they get smothered from some of that kind of input that might happen. That could be an acute event. And then we have something else to consider where there's a little bit of research that's happening with more of these chronic chemicals that are persistent in our waterways, and some of those are going to be your heavy metals. They're not quite yet for mussels, but some of those emerging contaminants that there's a lot of interest being generated with, like PFAS, some of those kind of legacy chemicals. Mussels are just now, starting to be researched on how does that affect them in their life cycle, because essentially what they'll do is they might not kill them right away, so they're going to bioaccumulate into their tissues and into the shell material. So on one hand, they're removing some of those contaminants and chemicals, pollutants out of the water and storing them, or they'll have a slow release of them. So ultimately that might affect their growth and their longevity. So they might not live as long. They might not be as robust, or being able to reproduce at an amount of what they used to do, if it's affecting that kind of disrupting their reproductive cycle, for example. So it might not kill them immediately, but we could see these chronic effects. I don't know if that's a long answer to it's not quite a straightforward answer, if you will.
All right, thank you, Sarah. Are there more questions in the chat?
Yeah. Are the mussels tested for forever chemicals like CH8?
So the question was, are the mussels tested for the forever chemicals? Not for our study.
Yes.
I'm sure there are other. Yeah,
Sarah.
Sarah just mentioned the PFAS, and that's starting to be studied. We are not doing that in our study, though.
And then is there a reliable species or a reliable identification key to help ID the mussels, or are volunteers relying on an on-site expert to ID?
So our volunteers for identification are taught to ID while they're at the event, but for a lot of the people, they're repeat volunteers, so they kind of gain more information. But then we also have people like Sarah and Bruce who are kind of around, so when they get stuck on an identification, they can help out.
Similar studies, or would you any point to a similar study further south here, like here, Louisiana, so close like the dead zone, or I guess maybe a more complex chemistry down there, like anything like that?
So the question was, are there similar studies happening farther south, like in Louisiana? I don't know the answer to that question. Sarah, I don't know if you know of studies outside of Illinois, or if you can answer that. I'm sure there are studies happening. That's as much as I can say on that.
Can you repeat the question real quick? Sorry, I was answering one of the chats.
Yeah, the question was
studies where
if there's similar studies happening down south, like in Louisiana, in the dead zone.
Oh, for like you mean like long-term
you
monitoring studies? For that, you mean?
Yeah.
I'm not aware of long-term monitoring studies like in Louisiana, not per se like ours in some of the rivers there. I know that there are other long-term studies that's happening that has happened in Tennessee and some in the Southeast. And I'm trying to remember if there's some on the Pacific Northwest. And in terms specifically of the Gulf, that would be more marine based. I'm sure that there's some long-term studies of different taxa groups, and maybe some of the marine mussels. I don't know. I couldn't even tell you as far as like, thinking about any kind of long-term studies on oyster beds. That would be around that area in the Gulf specifically. And as far as the river systems to the Gulf in that lower part of the watershed, any long-term mussel studies I'm not aware of.
Thank you.
Okay. I've seen some videos on Instagram on captive breeding and releasing of endangered mussels near Chicago. How does that process work, and does it work?
I do know we recently had a trip to, was it the Urban Streams Research Center, which is in Warrenville, near Naperville. And I know they have a mussel breeding program. And we got to tour it. I don't know if I know much more than that. I can't remember. Do you remember which mussels they were doing?
I can answer that question.
They were working on 5 different species while we were there. And from what I remember, they had like these indoor tanks that had all the baby mussels in them. And then when they got to a certain size, they would transfer them into these tanks that they were housing outdoor in their nearby waterways. And then once those get to a certain size, that is when they will release them. But I suggest looking into them because I think their website probably has a lot of information about what they're doing.
Yes, that's correct. They have some good summaries of what they're working on. And right now they're only working with our common species. So while all mussels are protected in Illinois, right now they're not specifically working with any of the state listed threatened or endangered. So one of the things that they're promoting, which is awesome, is to keep common species common. And we've lost a lot of those common species anyways out of Chicago region. So they're trying to build back this community of mussels in Chicago, one that'll help then hopefully in the future pave the way for being able to work with more of these listed species. And yes, they have several species that they're working with right now. Primarily they've worked with plain pocketbook and fat mucket, I think, as far as a couple of the really common ones. And then they worked with fluted shell, and they had Wabash pictio, which was a great new species that no one else got propagated. They were the first ones to propagate that. So they'll have fish hosts in their system. And then exactly as Hannah had described, is kind of their general process.
Okay, we are getting close to 8, but we'll still take a few more questions. Yeah.
I take it these have to live in running water, not in a pond?
So the question was, do they have to live in flowing water? I am pretty sure there are also mussels in lakes. It is not what we study, so I don't know.
What happens to them in the winter when it freezes?
So the second question is what happens in the winter when it freezes. So a lot of the time, especially if water gets a full sheet of ice on it, the water underneath the ice is actually warmer because of the insulation from the ice. And then they are also buried in the sediment. So I imagine that the sediment can also have a different temperature than the water. And other than that, I don't know if they have any more adaptations to survive through the winter. What was that?
Are they edible?
And then the next question was, are they edible? I would say technically, yes, you could eat them, but you probably wouldn't want to.
Yeah, for sure. So
what does eat them is any streamside mammals, for example, like a muskrat or a raccoon will eat them. Maybe like a snapping turtle probably does. Birds eat them. I don't know if fish do, really. Probably not. They don't have a system to open the shell. That would make sense.
Yeah, drum will eat them. There's a few fish that will munch on them. And black carp. Everybody get that on your radar. To answer the question for the winter, some quite a few species actually will bury inches deep.
Every rain, the water's eating the stream bank severely. I wish to stabilize the stream banks so the soil doesn't go into the Gulf of Mexico. What do I do?
So this question's about stabilizing a stream bank, plants, repairing vegetation is really important for stabilization. I think, and again, I don't know too much about this, but I think there's like some uses of like rock and stuff like that that you can build into the stream bank to help stabilize it. I don't know if you have anything to add to that, Danielle.
Yeah, just, I mean, it's very situation oriented, you know, he talks about a stream that spikes, but like, if it's a truly urban stream, then there's probably not much that an individual landowner can do. If it's, you know, a little bit more like very suburban, you know, not high density, and it doesn't come up more than a couple of feet for most storms, then vegetation is probably the best bet.
Did you guys hear Danielle's answer? If you could answer in the chat if you heard it. Do you have a question?
I was just gonna add, I think if he's looking for resources or contacts, this local soil and water conservation office would be a great start.
I'll put that in the chat.
Okay, cool. Are there any more questions?
How I signed up to be a mussel grubbing.
So the question was, how do you sign up? So the volunteer sign up comes out of mostly Upper Sangamon River Conservancy. And you don't have to be local to do it, but just know if you're coming from Alton, where we are, it's about a 2.5-hour drive to go there. So I would say the best thing to do is they probably have newsletters that you could sign up for, and or follow their Facebook so you can see when it happens. The events happen usually in August. So,
Follow our Facebook.
or you could follow our Facebook. Yeah.
We posted too, because we're going.
Yeah. Okay, I think I'm gonna wrap it up now. If you guys have any more questions, you can feel free to send them to me. I believe my email is connected to the email that you got about the Zoom link. So you can email me or Danielle or RiverWatch in general. And if I can't answer it, I will send it to somebody who can answer it. Thank you for everybody who attended virtually and everybody who came out tonight. Since Bruce never said anything, Danielle is nominating him to also say something now, if you would like.
Yeah, I'll just say what an honor it is to work with all the great people at RiverWatch and with Sarah. Sarah's the one that got us hooked on going out and doing mussel surveys to begin with. She came and gave a presentation to our group in 2011 and hooked us up with Steve Buck, who's a researcher. And so we got interested there. And then when we wanted to step it up a notch, we got Danielle involved, and Danielle and Hannah have been great. So that's all I've got to add.
All right, thank you, Bruce. Thank you, Sarah, for also answering questions. And again, thank you everybody for attending. If you missed the first slide, I don't know.
February 9th.
Okay, the next speaker series is February 9th. And you can find more information about that on our website or on our Facebook page. It's going to be about dragonflies. So I feel like a lot of you will also be interested in that. So thanks again for attending and have a good night, everybody.